So good morning, everybody. It's 10 o'clock and we shall start. Welcome to the second day of the conference of the Baltic Conference on Intellectual Cooperation. And mathematics for society is really very important, as we also have mentioned yesterday already. This was a language that the world is described. Whether it's exactly like that, we don't know yet. But nevertheless, yesterday we've been analyzing what is the role of mathematics in policy. We have been analyzing the modeling of pandemic, and today we shall analyze more general problems. We have got all this before the final remarks and before the lunch. We have got 6 talks, and I would like to ask all the speakers just to keep the time, because it's 20 minutes for every talk, including a question or 2. But well, let us try to really to keep it. And we shall cover really very broad areas, starting from the general problems of complexity, and then the very intriguing problem of the quantum computing, and then going to the modeling of biostructures, and then water resources, and then we turn our look to the history telling that the navigation also needs a lot of mathematics, and it was understood long time ago already. So let us start then, and the very first speaker is Professor Juhan Aru from the École Polytechnique Fédérale de Lausanne. He has been studying in Cambridge and Lyon and been working in Zurich, and he's also the member of the Estonian Academy of Sciences. So that about the universality, when can complex be simple? And for all the speakers, actually the time is also going over there. You will see it on the top there. So, Johan, the floor is yours.
Thank you. Good morning, everyone. First, my thanks go to Professor Engelbrecht and for inviting me. It's always a great pleasure to talk about mathematics, especially in this beautiful setting. And it's important to remind to mathematicians that you also have to think about society. This is not an immediate thing. So what I would like to tell you about today is universality, or at least one aspect of it. And the basic question I have in my research, or my research is also my life, is understanding how these beautiful pictures emerge. Because we know from natural sciences that they are not being brought to us in a full shape. They are actually formed by microscopic interaction, formation processes. We already heard about this yesterday. The question is, how does macroscopic structure emerge from microscopic detail? So this is the basic question, and today I would like to address one aspect of it, which is called universality. So this is the question of why do many structures we see look similar? Why do we see fractals everywhere? Why do we see statistical self-similarities? And so on. And I would like to just give some brief examples on that. So let me start still from what has occupied probably the minds of all of us for a year or so, or occupied, taken over, etc. So this is of course, the corona crisis, and these graphs are from the very beginning of the pandemic, and I usually don't do too many plots. I'm a pure mathematician, so these plots are not as beautiful as you saw yesterday, but I was quite excited to see them. So I even think that this reminded me of being on a stormy sea where you feel unwell, but everything is beautiful around you, these big waves, etc. And here it was a little bit similar. I was anxious about the world on different scales, but then I saw these beautiful exponentials. So here on the y-axis, it's the log of the newly reported cases, and on the x-axis, the time. And this had a certain soothing effect, I think. And we already heard yesterday that, there was a certain disagreement. Is it a complex process, this spread of the pandemic, or is it simple, as we heard in the last talk? And I have to say that I agree with both, also in the spirit of yesterday's dinner. But first of all, I would actually say that for me, most of all, this was surprising. It's maybe a naive thought, but I was surprised to see such pure exponentials, and I was surprised to see that the exponents are so similar. So here we see these really purely linear things. Of course, behind it there is a complex process. We heard there are many levels of details when the pandemic spreads further, but even in the initial stages, you can think that, the way we spread the disease is basically one by one, or a little bit by aerosols, a little bit by contact on the surfaces, but everyone behaves differently. Some people hug, some keep always distance, some live together in groups, some are solitary, but it spreads so that in the end we see basically a nice exponential curve that is characterized by one simple exponent. And for me, this is, at least for me, my naive view, this is simply fascinating. Of course, there is a mathematical understanding of simpler versions of that, and then I would like to talk about one such thing that fits into picture of universality. So this is in the very initial stages. You can use a very simplified model of branching processes that stems from already the 19th century, and back then it was used actually to study whether the family names will survive. So then that was an important thing, will my family name survive or not, and branching processes were invented to describe that. And branching processes are very simple. You start with one individual, then you say that this is, my first infected person, and it gives the infection to some random number of new individuals. And each of these individuals further gives the infection forward to some random number of individuals. It could be none, because the guy just sits in a cave. It could be many if they go to a football match. And so each individual infects a random number of individuals. If you make some assumptions, for example, if you say that all these numbers of how many people in fact are independent, have the same distribution, you see a very simple phenomena. There is this one parameter, which is the expected number of individuals that are infected, that governs the whole dynamic. If this is smaller than 1, then we see that the whole tree dies out exponentially. If it's bigger than 1, it will grow exponentially. And if it's equal to 1, you are in the critical case, which is even richer. I say that this is an example of universality because here you see the individual distributions can be different, can be complicated. It could be that you just give rise always to 2 other infected individuals, or with half to 3, with half to 1. But the macroscopic behavior we'll see is exactly the same. Okay, so it doesn't matter on the individual details of xi. Also, the conditions can be smoothened, but you always see this macroscopic either exponential growth, dying out, and even these parameters are very simply defined by just this one expectation. Okay, so this is a basic example of universality on microscopic level. If you look on the individual level, there is a complex process, but at large, what we observe, it becomes simple. So I would like to give you some more examples of this universality. So first of all, universality of caps, and here is a morning question to you. So what relates neutron scattering of heavy nuclei? So you shoot heavy nuclei with neutrons, and you see at what frequencies they scatter with large amplitudes. You look at zeros of the Riemann zeta function. So this is a number theoretic function that encodes the behavior of prime numbers. So you look at them on the so-called critical line. And then you look at bus arrival times in a little village in Mexico, Cuernavaca. So, what could relate such things? Okay, so here are the graphs. So here are the graphs. So there are the histograms of the gaps between these resonance frequencies on the left, between zeros of the zeta function in the middle, and between this between zeros on the right and bus arrival times in the middle. And you see that I have fitted some curve. Actually, not me. It's I should have said it before. Most of these figures come from the original scientific articles that I would be very happy to refer to. But you see that there is a curve that fits these histograms very well. So what does this curve stem from? It comes from random matrix theory. So what are random matrices? These are just some tables of random numbers. So as you see, you just put together a table of random numbers, and then there is some set of numbers that characterizes its nature. So this is called the spectrum, and up to symmetries of this matrix, it characterizes its being. So this is just some set of numbers related to this collection of random numbers. And it comes out that this set of numbers, when you make your matrices bigger, bigger, and bigger, starts having a universal statistical behavior. You can look at the histogram of this set of numbers. You can start looking at the difference between them. And exactly these differences are the ones that give us these theoretical curves fitting the histogram. So you take a matrix with Gaussian entries, independent, you look at what it's called its spectrum, and then you discover that the statistics you see is the same as for a number theoretic function, a deterministic function, for bus arrival times in Cuernavaca, and so on. So here we see there is universality even on two levels. First of all, in this random matrix theory, there's a lot of freedom. Again, I have these random numbers, but I'm not telling you much about them. I'm not telling what is their exact distribution. Are they just taking values plus minus 1? Are they taking values on the real line? So there is universality there. But then we see that it's actually the same behavior we observe in systems that have nothing to do with matrices a priori. And this is a very, very active area of research at the moment in probability theory with lots of wonderful stuff. But okay, you might say you are still not happy with what I'm saying. I'm just telling you something about distributions, random numbers, that's not, maybe that's not your thing. You want to see more pictures. So let me show you a few more pictures. So the domain I am working in is at the moment two-dimensional statistical physics. And there are many models there. For example, there is the model of percolation that was invented in 1950s to model actually the flow through gas masks. So it's flow through porous medium, and on this image you can think that, okay, I'm looking at a very thin material, and I have these hexagons are representing pores, and if it's black, I have the water or gas cannot pass through, and if it's white, it cannot. And every pore is maybe decided to be open or closed with some coin toss. A second model is the Ising model, which comes from the beginning of the 20th century and basically studies how the spins in a thin sheet of, in this case, two-dimensional case, in the thin sheet of ferromagnet align in a magnetic field. So the pluses like to actually be together and minuses as well. And the third one is a model of the self-avoiding walk, which was invented to study long polymer chains. So polymers are put together from single molecules, and you ask what shapes can it take, what are the statistical properties you can see at large. So again, a question for you, what relates the above models? And I must say it's probably not so simple to see if you look at these pictures, but what if I show you these pictures? So you can think of these pictures as they are just being taken from very, very far away. I have taken the same models, but on a very large scale, and at what is called the critical parameter. And then you see that there is some geometric similarity here. So can we get a grasp of that? Well, maybe not us, but it was done already in the end of 20th century by Otto Schramm. So he made a key observation, which is that in these models, all of these models share something similar, which is that they have these interfaces between, for example, blocked and open pores, or plus or minus spins, or just this walk itself. And these interfaces can be explored in a certain local way, and they are expected to satisfy some symmetries. And he realized that if you take these things, some local constraints, some statistical symmetries, then this actually gives you very few options for describing the process. So he actually understood what is the stochastic process that should describe these interface curves that you see between black and white. And now they are called Schramm-Loewner evolution. It's a one-parameter family of universal processes that describes all two-dimensional statistical physics models. So this is one of the rare cases I know where pure mathematics actually went back to the lab, in the sense that experimental physicists started looking for these curves in their experiments, trying to understand whether they appear and with what exponents. So I thought that I would also like to tell you about some principles behind universality, but this, I have to warn you, is not something I'm actually capable of doing, because we don't know. We don't know in these more complicated things. And in fact, yesterday morning when preparing, I understood that just an article appeared on Archive which said that, look, probably this universality for a concrete model of percolation is much more widespread than we think. Okay, and you know, the only thing I can do, I can tell you a little bit in the context of a very well understood example, which is the central limit theorem. So this is probably something you have all heard about, which is that if you start adding up independent numbers, then under certain quite general conditions, your distribution will start looking like the bell-shaped curve, the Gaussian distribution, and if you normalize it well to be of unit scale, it actually converges to the Gaussian law. So here again, this is an example, a prime example maybe of universality, because the exact laws of these individual numbers do not matter. So they don't actually have to be the same, you just ask from them that they are not too big and they are independent. And we see also the phenomenon that we have seen in the previous slides, that there is some universal law, some special law in the limit. Okay, and here from this we can see some principles that actually seem to be present in all these models. One is that if we want to have a universality in a complex system, then all the components need to really have a negligible contribution. Otherwise, you know, if there is one big one, it will dominate the sum. Second, we need to have independence or local interactions. Otherwise, again, if there is one factor that influences everything, it will influence the sum. And finally, what we see is that the emergent universal objects are often very special. So here the Gaussian law has many special properties. It has maximum entropy, it is stable, and so on. But this is also the case in these more complicated cases I was mentioning. So this brings me to the end, where I would like to draw some conclusions and two little thoughtlets. I wanted to explain to you that simple behavior can arise from complex systems, simple universal behavior. And I think this is kind of interesting, because it means that even if you have complexity, you could be predictable and controllable. So if you think that this branching process perspective of the spread of the disease, then it's very clear that policies want to act on this R0, this spread coefficient, and they act locally by putting constraint on individual spread, how each individual spreads. And you can do it by vaccination, you can do it by some masks, etc. So this is one thing. So in that setting, I think it's interesting to think about when does this universal actually emerge, this universal behavior, and what are the limitations. When we understand this better, we can maybe also start understanding in which settings of society, etc., they could be applied. And finally, the thoughtlet is that, you know, in this world we have seen it's hard to avoid complexity. Systems get more information, more entropy, it's hard to avoid it, but maybe one can steer it towards complexity, towards simplicity, if we know which complex systems have a simple macroscopic behavior. I think it's possible that you can still, you know, with policies, laws, steer things towards complex systems where you have this predictable process from microscopic to macroscopic. So this was just a question for you, and I thank you for your attention.
Thank you very much. I think that it's really very important to come with sort of an analysis from the principles, and as yesterday, you know, as explained about proof of physics, it's exactly the same sort of complex structure. And I think a model actually was described in this very first treatise, about you know, the beauty of fractals, as you remember, that was published in the 80s actually, that's all that. So I don't see any question just now. Later we can carry on the discussion. So thank you, Johan, very much indeed. And now we come to the mathematics of quantum computing. It's Professor Andris Ambainis. He's from the University of Latvia, Faculty of Computing Science, and what is very important today,
You have got once also the grant from the ERC, which is a quality mark indeed. So, Andres, floor is yours.
Thanks. So I'm a mathematician working in quantum computing, essentially. Computer scientist by education, mathematician in my heart, working in a field that is largely dominated by physicists. So here is what quantum computing is about. Quantum computing is an emerging model of computation that attempts to compute via quantum physics. Information is encoded in quantum states, for example, spins of atoms, and one computes by manipulating quantum states. And as you might have heard from popular press, quantum computers may be much faster than conventional computers if they are built. In the best case, if we build a quantum computer, it will be able to solve some problems that require exponential time for conventional computers. So it would push the boundaries of what is efficiently solvable. And there are some small-scale quantum devices that are appearing. Quantum computing is an extremely hot field of science and technology. Every major IT company of the world is trying to produce a quantum computer. Here are some devices that have appeared over the last few years. All of those devices have received a lot of publicity. All of them are quite small-scale devices. Google's device is probably the most advanced one, and it is right outside the boundary of where this device could be modeled on conventional computer. So Google's device is just big enough and can just run for just long enough to make it unsimulatable on a classical supercomputer. It can't yet solve anything useful, but it's impossible to simulate it even on a very expensive conventional hardware. And the hope is that as these devices grow more powerful, they would be able to solve problems that are hard for conventional computers. And one of the biggest possible applications is modeling physics and chemistry. On a mathematical level, chemical reactions can be described by differential equations. And we know those equations, but in many cases, solving them on a conventional computer required either very large computation time or very large memory. And quantum chemistry is a very substantial application for high-performance computing. And if we built a quantum computer, we would be able to model chemistry more efficiently and more precisely. And ultimately, modeling chemistry could lead to many advances that affect many fields of life. The prototypical dream of computational quantum chemistry is to come up with better understanding of processes, how nitrogen gets captured by plants, and to come up with better processes for producing nitrogen fertilizer industrially. And if this dream came true, it would save a couple of % of world's natural gas, with all the consequences to reducing global warming. So this is a big motivating dream. And then the hope is that quantum computers could be a part of solution here. And quantum computing is on the boundary of several sciences. And typically one thinks of it as a field between physics and computer science. But mathematics is also here, and mathematics actually plays a very important role. So by training, I'm a computer scientist. I'm a theoretical computer scientist, which is a sort of mathematician working in computer science. I don't actually have a degree in physics. My quantum physics education consists of reading Feynman lectures on physics, on quantum physics, when I was a graduate student. And yet, I am capable of working in quantum computing and inventing methods of computation for quantum computers. And what happens is that in our field, computer science and physics communicates through mathematics. Mathematics is the intermediate layer between these two sciences. And as a result, physicists can build quantum computers without really understanding what algorithms will be run on them. And I can understand, and I can build algorithms for quantum computers without understanding every detail of physical device. Yeah, so in essence, quantum mechanics is a mathematical theory that can be described by about 4 or 5 mathematical postulates, which say that the state of a quantum system is a vector in Hilbert space, the evolution is described by Schrödinger equation, and so on. And these 4 or 5 postulates, the number depends on the particular presentations and the particular book on quantum mechanics. They describe a very simple and very beautiful mathematical model. And this mathematical model describes countless quantum systems, whether these systems are atoms, photons, or something else. They all follow the same mathematical framework. So in a sense, quantum physics is a physical meta-theory that describes mathematical principles for a large number of physical systems. So here's a quote from my famous colleague Scott Aronson, who was at MIT at that time. So what is quantum mechanics about? From his and also mine perspective, it's about information and probabilities and observables and how they relate to each other. And so what quantum computing and quantum information does is it builds a model of information following quantum physics. So in the conventional quantum physics, one studies particles and variables such as location of a particle, which are real-valued numbers, and their state is a vector or wave function in an infinite-dimensional vector space. And what quantum computing and quantum information does, it takes the simplest possible mathematical version of this. It takes a quantum system with just 2 basis states, which is now called quantum bit, and it attempts to build a model of computation based on these simplest possible quantum systems. And then there are many ways how one can implement a quantum bit. I put 3 different devices based on 3 different technologies on the left. The top device is the most famous one. This is Google's quantum computer, which is based on electric current flowing in a superconductor at extremely low temperatures. So quantum state that encodes information is electric current in this superconductor. The middle one is a quantum computer based on trapped ions and levels of energy in trapped ions. It's a device produced by American company called IonQ. And the bottom one is a Chinese device, which they call desktop quantum computer, which is a demonstration device where quantum information is encoded in nuclei of atoms and spin of atom, which shows how atom, which is a variable that describes how atom behaves in a magnetic field. And what happens is that in these 3 devices on the left, there are three completely different physical systems, but they all implement this basic functionality of quantum bit. And I, as a computer scientist, I use this basic functionality of quantum bit to solve various problems in computer science. And so in this picture, physicists work with the things on the left, computer scientists work with the things on the right. In the middle, there is a mathematical model of quantum bit that allows physicists not to know about computer science and computer scientists not to know too much about physics. And on the right, there are three examples of things that one could do with a quantum computer. One would be able to factor large numbers efficiently. So given a large number that is a product of two smaller numbers, we would be able to find these two numbers more efficiently, which would have implication for security of some cryptographic schemes that are used all over our everyday life. We would be able to search in large amounts of data more efficiently. This is the middle picture, the phone book. And we would be able to compute logic formulas more efficiently with applications to all areas that can be modeled by logic formulas. So the first two examples on the right are the classics of quantum computing that have been known for more than 25 years now. The third one is something to which I contributed a bit myself in mid-2000s. Yeah, so there are different ways how mathematics comes up in quantum computing. So mathematics serves as this intermediate layer between physics and computer science. One can invent quantum algorithms for mathematical problems. It is possible that mathematical problems in, say, fluid dynamics or whatever else is modeled by differential equations would be among the first applications of quantum computers. Mathematics can serve as a tool for developing quantum algorithms, and mathematics can serve as a tool for understanding the limits of quantum computing. And these two last items are subject of my own research, and in the last 6 minutes, I would like to present an example of one of each from my own work. So first, how do we use mathematics to develop algorithms for quantum computing? So here is an example. There is a computational task called element distinctness, in which one has a large array of numbers, and the task is to find equal numbers in this array. So this is something that can be solved by quantum computers faster than by ordinary computers, assuming quantum access to this array. So if there are just two equal numbers and the array is of size n, then conventional computers would need roughly n steps to find the two equal numbers. They would have to look at this whole array, and quantum computers would be able to do faster in roughly n to 2/3 steps, assuming that they are provided with quantum access to this array. So for an array of size 1 million, that would be 10,000 steps instead of 1 million steps. And the way this works is by using a paradigm that can be described as a search by random walk. So we first think of a conventional algorithm that tries to find the two equal numbers by moving randomly on this array. And this algorithm remembers subset of this array, and then it randomly adds and removes elements until this set contains two equal elements. And then we take this random process and we quantize it. We replace random walk by a corresponding quantum process called quantum walk. And then it turns out that one can show a general result about improving such algorithms, that if under certain conditions on the random walk, we have a random walk algorithm that finds solution in t steps, then we can produce a corresponding quantum algorithm that finds solution in square root of t steps. An example of this is this algorithm for finding two equal elements. But in the following years, this method has been generalized far beyond that, and it has become a very substantial tool for designing quantum algorithms. And now, second example, quantum computers can solve many things. So how do we know that there is no faster quantum algorithm? So we know that I showed this example that quantum algorithms can search arrays faster than conventional algorithms. Is there an even faster algorithm than the one that I developed or not? And in some cases, we can actually prove that there is no faster algorithm, and whatever we have invented is the best possible quantum algorithm. And the model, to prove such results, we need model. Every impossibility result that something cannot be computed applies to some model of computation. So the model is that we have some task that depends on input data, and input data is given through a black box that answers questions about values of variables. And we can access this black box quantumly within a quantum algorithm. And what we count as the time complexity of algorithm is how many times it accesses this black box. And once we go to this framework, we can not only invent quantum algorithms, we can prove that the quantum algorithms that we have invented are the best possible. And to analyze quantum algorithms in this model, we look at how the state of a quantum algorithm changes throughout this algorithm. And we can describe quantum state as a vector in some vector space. And then this vector during the computation, it starts to depend on the input data inside this black box that answers questions about values of input data. So the key question here is how do the elements of this quantum state depend on input data? And one can show that, in fact, this is a pretty simple mathematical lemma, actually. The proof is only a few paragraphs. One can show that after accessing input data k times, the state of a quantum algorithm can be described by polynomials of degree at most k. And then, in some cases, we can even establish an equivalence between polynomials and quantum algorithms. So there is this result which we showed a few years ago together with Scott Aronson and a few other colleagues. But if a problem can be solved by accessing input data by a quantum algorithm that accesses input data just once, then we can approximate this problem by a polynomial of degree 2. And if we can approximate it by a polynomial of degree 2, we can turn this polynomial into a quantum algorithm. And this relies on an inequality in the functional analysis by the famous 20th century mathematician Alexander Grothendieck. Thanks.
Yeah, thank you very much indeed. We now know a little bit more about the mathematics behind this complicated this computing. And I have a very short question. Is it possible that the next level quantum computer will be built in Latvia?
No.
Why not?
Building a quantum computer is now getting extremely expensive. So.
Okay. Any more short questions? Yes, please. Yeah, Johan.
Hello. So
Thank you for the very nice talk. I have a very naive question. So I remember that there is a mathematician, Jill Galai, who always says that, quantum computers in the end will not be useful because there will be too much noise in the system. So what is the status now? Has it been proved that he's wrong, or he's also on the blog of Aronson, always commenting and...
Well, it's hard to prove him, well, I think he's wrong. It's hard to prove him wrong in a mathematical sense, because what he has is not a precise argument, but I think he's making wrong assumptions.
Yeah.
Thank you.
So thank you very much. I think that we have now to go on over. Thank you. And now we go over to biostructures. And we have two talks here, and the first one is by Dr. Czeslavas Venslavas from the Vilnius University Institute of Biotechnology. He's the head of the bioinformatics laboratory and also the member of the Lithuanian Academy of Sciences. I do hope that you, oh, thank you very much, so that he's already there. So the floor.
Hello. I hope you can hear and see me. So, as the chairman already introduced, I'm going to talk about computational research in protein structures. And let's maybe start from the essentials. So basically, we all know that every organism can be characterized by genetic information, and this information is stored in genome. But this genetic information would be just silent if not for proteins, because proteins turn this information into action, because they perform nearly all biological functions. And so what is a protein? Protein is a specific sequence of amino acids connected in a linear chain. So basically, it can be represented as a string of letters. But they don't exist as a string of letters. They assemble in specific three-dimensional structures. The structures can interact with each other, forming complexes and even entire protein-protein interaction networks. So in a sense, protein world can be compared to a language where proteins correspond to words, complexes to sentences, and interaction networks to stories. And this is not just a nice analogy, because as I will talk about this, some methods actually are common for protein research and language research. But in order to understand these stories, we need to know three-dimensional structure of proteins and protein complexes, because that is their functional form. But there is a small problem that determining protein three-dimensional structure is expensive, time-consuming, it is not always successful. So here is the data. Currently, there are about 200,000 structures known, and this has been accumulated over more than 50 years. And if we compare how many sequences we know now, it's more than 200 million. So as you can see, structures only correspond to a tiny fraction of known protein sequences. And this gap is increasing, constantly increasing. So it's clear that only computational methods can bridge this gap. And in general, there is well, in theory, it's completely doable, because protein 3D structure is determined by its sequence, and it's known that folded native structure corresponds to energy minimum. So in principle, it should be possible to compute protein three-dimensional structure using laws of physics. And we know that nature does it in seconds or sometimes even milliseconds. However, the problem is that the protein chain, unfolded protein chain, can assume an astronomically large number of different conformations, and energy estimation is not accurate enough. So it's even for a small protein, it's a problem to compute this three-dimensional structure. And there are very many different approaches for predicting protein structures. Some rely on principles of physics. Some also include evolutionary considerations. But most methods, at least until now, they were using went through two major stages. One is to construct a set of models, not a single model, but a set of models, and then select one that presumably would be most similar to the native structure. And this would be a final model. Now, with every approach, you need to know how well it works. And it's important, and for this, you need to have an efficient method to compare model with the native structure. And although this seems like a trivial task, I can assure that it's not, because protein structures are really complicated and many things in them can be compared. Of course, model selection is even more important step, because it's not only important for method development, but also for practical use, because if you have a set of models generated by very different methods, you would like to select one that is closest to the native structure. And next, I'm going to present methods developed in my lab that actually address these two problems: comparison of reference structure with models, and also the other problem for model selection. And here mathematics come in, because the recurrent theme in my lab is actually use of a mathematical construct called Voronoi diagram. And I will tell you why. But first, what is this Voronoi diagram? Voronoi diagram was introduced by Georgy Voronoi, mathematician. Some considered Russian mathematician, some Ukrainian. Nevertheless, he was a brilliant mathematician, and he introduced this diagram in the beginning of 20th century. And this is a way of dividing plane according to some objects, in this case, centroids. And if we have a centroid, so we can assign all points that are closer to this centroid than to any other centroids to the region that is called Voronoi cell. And if we do this for the entire plane, we get a classical Voronoi diagram. One nice property of this Voronoi diagram is that it unambiguously defines neighbors. And this diagram can be built for spheres in 2D and also for balls in 3 dimensions. And this is exactly what proteins are. These are atomic balls, sets of atomic balls in 3 dimensions. And the Voronoi diagram of balls provides neighbors for every atom, also provides interatomic contact areas, and also takes into account structural context, which is not easy to do with distance-based methods. Now, in nature, there are a lot of patterns that can be modeled by Voronoi diagrams. And also people use Voronoi diagrams for very different things, like building maps or looking for closest airports and so on. So now I will tell you about the method that we developed for comparing reference structure with the model, and more generally comparing two structures or two surfaces. And the method is called contact area difference score, or CAD score. So the definition is quite simple, and it is based on contact area difference between pairs of interacting residues from one structure and corresponding residues in another structure. So we can calculate absolute contact area difference, and then for the entire structure, we can sum up all these differences normalized by total area of contact areas, the total sum of contact areas, and we get cut score, which ranges from 0 to 1, where 1 defines all, means that all contacts are identical in two structures. Now, how it works, here is an example. So we have two structures, native structure and a model. And probably you can see that initially it's difficult to say whether these two are similar or not. But cut score says that they are reasonably similar. But if we split this structure in half, here where the correspondence is worst, and superimpose these pieces, so we can see that this part and this part actually for these two structures are very similar, and cut score actually shows that. So basically, cut score can provide both balanced comparison of structures and also pinpoint local differences in structures. Now, since cut score works with surfaces, it can be used to directly compare surfaces such as protein-protein interfaces, in this case, interface of protein dimer. And if we have two models, we can see that this model has a better predicted contacts than this one, and cut score clearly reflects that. So based on these nice properties, cut score has become a standard measure in the field and is used in infrastructure, computational infrastructure in several organizations, for example, Protein Structure Prediction Center or Cameo. Now, the second problem that I was telling you about is selecting the best model or structure without knowing the answer, without knowing what the real structure is. And for this task, we developed also Voronoi tessellation-based approach called VoromKA, which stands for Model Quality Assessment. And the idea is also similar as to cut score. We use Voronoi tessellation, but in this case, we collect statistical data from experimentally determined structures, and we can, based on this data, we can assign pseudo energy for a pair of interacting atoms based on Voronoi diagram of balls. And in this way, we can assign these pseudo energies for interaction with all the neighbors, and then transform all these pseudo energies into score from 0 to 1. And we can do this for atoms, then for residues, and for entire protein structure or protein complex. Since VoromKA works on surfaces, it can also directly score interfaces, protein-protein interfaces, which is very useful in protein research. So again, example how it works. So here we have 3 structures of the same human protein complex called 911. And these 3 structures were solved by 3 different independent labs. And you can see that one of them actually shows much lower VoromKA scores. And if we inspect the structure, we can see that this structure is incorrect. So basically, it's not only VoromKA can not only select the best model, but also check whether the experimental structure is correct or not. Also, VoromKA can be used directly, as I said, to directly assess protein-protein interfaces, and in this case, to distinguish biological assembly from the one arrangement based based on forces of crystal packing. Most recently, we actually extended this Voronoi tessellation approach and combined it with deep learning. So we transformed Voronoi tessellation into a graph, protein represented as a graph, and then used graph convolutional neural network trained to predict the residue quality of the structure. And this method is an improvement of the original VoromKA approach, and also one of the best currently available. Now, it's always important to check how your methods work and how they compare to other methods developed currently. And in protein modeling community, there are two experiments, two community-wide experiments called CASP and CAPRI, and they operate in test blinding mode. And how they work. So basically, the organizers solicit sequences from experimentalists, sequences of proteins or protein complexes that either are solved but not published yet or close to being solved, and pass them to teams of bioinformaticians. And bioinformaticians are then given some time to generate models of individual proteins or protein complexes. And once the experiment is over, then independent experts compare models with native structures, and based on that, they decide which methods work best, and in general, what is the state of the art in the field. So CASP looks at different aspects of protein structure prediction, and CAPRI looks only at the ability to model protein complexes. So we participate in these experiments, and one such experiment or combined experiment, CASP and CAPRI experiment, was organized last year, and our results were quite reasonable, but we were behind one other group, Baker group from University of Washington, and we were interesting to see, to check what was the reason. And we when we looked in more detail, we found out that actually the Baker group was better at overall structure accuracy, which is mostly determined by the accuracy of individual subunits. But in our case, but our results were better according to subunit interface accuracy. And we believe that the main reason was the efficiency of Voronoi, which we used to select actually the correct interfaces. Now here the results, I presented the results on modeling protein complexes, but the most impressive results were obtained of prediction of monomeric protein structures, where Google company DeepMind participated under codename AlphaFold 2, and they were able to produce models that actually were almost the same accuracy as experimental results. So based on their results, actually many experts in the field consider that the computational prediction of monomeric protein structure is essentially solved. So what was their approach? Actually, not much is known about their approach because it's not yet published, but we know that they used a deep learning system with attention-based mechanism, which is borrowed from natural language processing. They consider proteins as a spatial graph, and their system is end-to-end system. So they start from sequence and end up with three-dimensional structure. Now, the question, is this the end of computational protein structure prediction, and should we look for another job? Probably not yet, because although monomeric structure prediction can be, monomeric structures can be predicted quite accurately, the process of three-dimensional structure assembly from sequence is still not really understood. Also, there was no such breakthrough in prediction of three-dimensional structures of protein complexes, and this remains a challenging area of research. Also, probably artificial intelligence will have not so big impact of designing of new proteins. So I will switch now gears a little bit and tell that protein modelers also were not so standing aside and looking how this COVID-19 crisis unfolded, and they were actively participating, including our laboratory. And there were at least two initiatives where we participated. and these initiatives were community-wide initiatives, one organized by CASP Center, and that was selection of 10 viral proteins and predicting the models for all these proteins using very different approaches. And all these models are actually available for community, for experimentalists especially. Another initiative was from Capri organizers, and they selected 4 complexes that everybody modeled, and also these models are available in this repository, and hopefully both of these initiatives will help experimental research on COVID-19. So I'm coming to a summary, and I should say that mathematics and informatics play an increasingly important role in protein 3D structure analysis and modeling. Also, I think that a mathematical construct, Voronoi diagram, is really well suited for representation, analysis, and assessment of protein structures, and will be used more and more often. And hopefully, Voronoi diagram combined with deep learning may help to bring this breakthrough also in modeling protein complexes. And finally, I would like to acknowledge people in my lab, especially these 2 guys, Kliment Olechnovich and Justas Dabkūnas, who were essential in developing these methods that I told you about and participation in CASP and Capri. And thank you for your attention.
Yeah, thank you very much indeed. It's a very interesting talk about the biostructures and using the knowledge from the other fields like Voronoi diagram that, as you know, about the tessellation, which has been using widely also in material science, for example. And as you know, the recent Nobel Prize winner Roger Penrose has been also interested in tessellation, and it seems that it really opens ways how to combine the knowledge from the other fields also for a very complicated biological structures like the proteins are, and as we have witnessed from the slides. Just a short question. No? So thank you very much indeed for a very interesting talk. And we shall continue with biology, and the floor is given to Professor Mait Metspalu. He's a director of the Institute of Genomics, the University of Tartu, and this is the problems about genomes. So, Mait, floor is yours.
Thank you. I, of course, thank the organizers for inviting me. It's actually my second time in the Baltic Cooperation Conference. I was also lucky enough to be in Vilnius 2 years ago. Today, I have to say I'm not a mathematician at all. I consider mathematicians the Jedi masters and look up to them, and thanks to them we population geneticists can do our work using basic building on what mathematicians do. So today, my task I took is a little bit different than the usual talks. I'm not going to show you too much the actual results of what we do, but I wanted to give some insight into the intuition behind some methods that we use. By doing that, of course, there will also be some stories about the results. The title was Digging Genomes to Understand Our Past, Modern Tools for Old Puzzles. When I did the slides, then in some cases it went the other way around. It was old tools for new puzzles, but nevertheless. So what do we study? What do we do? Basically, we are interested in the genetic diversity of populations, and in our group, it's human populations. And the genetic diversity is shaped by three main groups of processes. First, it's demographic processes, or let's say evolutionary history. It's a complex array of events of population splits, population mixtures, migrations, which end up creating admixtures of populations that maybe have split a long time ago. So it's history. Then natural selection. When we think of humans, we left Africa, say 70 or 50,000 years ago, populated the whole globe with very many different environments, and naturally we had to adapt to these environments, and this definitely shapes the genetic diversity. And thirdly, especially in case of humans, actually culture can affect or change genetic diversity. For example, kinship structures in a population definitely have a say on the genetic structure of a population. So, and I would say that in most general way, it's maybe not the genetic diversity we are actually interested in. We're actually interested in these processes that shape genetic diversity, and we can study these processes by studying the genetic diversity. So first, so as I said, there are three main factors that change or affect genetic diversity, and I'll try to show examples on all three. Let's see if this will be possible. So first, a very old and very simple method that does not have anything specifically to do with population genetics, principal components analysis. It's more than a century old. To population genetics, it was introduced not so recently. But the trick there is, as you know much better than me, is it takes many characters that may be correlated with each other and creates new synthetic characters out of these, which then will not be correlated. So basically it shrinks the dimensions of the data set, makes it easier to see. And this is very widespread method that is used. Because mostly the genetic diversity of populations is shaped by the demographic history, we end up that more geographically more close populations are also genetically more close. And the principal components analysis reveals these patterns very well. Here we can see a principal components analysis of global worldwide populations. We see that the first principal component that describes most of the variation on the y-axis differentiates between African populations from all other populations, and the x-axis, the second principal component, discriminates between East and West Eurasians. Now, if we go into more detail, for example, we take European populations and we do a similar principal components analysis, and then we plot basically the results on a map. We can see that the principal components, the genetic structure revealed by principal components mimics the geographic map of Europe. Where these principal components become very useful is, for example, ancient DNA studies. Here we have, again, a principal components plot where when you look at the gray dots in the background, then these are modern European populations. I don't have a pointer, but the gray wave of dots on your right is populations of Caucasus and Near East, and then the gray area to your left is basically West European populations. Now the colorful dots are ancient DNA samples, basically from hunter-gatherer, European hunter-gatherers to early European farmers and to Bronze Age people. And not going into details, we can see how these how the genetic diversity or genetic patterns of these people that lived 10, 5, 3,000 years ago in Europe, how they are different from contemporary populations. And going into more details, we can, for example, consider the people who lived in Estonia, and we can do this. We can look at the same principal components plot, and now plot people who lived in Estonia on different time windows and see how they move on these principal components plot. I'm not going into details of how they actually move here, but this is the method that we can plot people from different time windows on the same principal components plot and see how the genetic diversity changes over time. Another method or group of methods that deal with genetic structure of a population, it builds upon a population genetics model called a Hardy-Weinberg equilibrium. The principle behind this is very simple. In diploid organisms, such as ourselves, we have, in our genomes, we have two copies of all genes. You can think of two alleles. Now, and these alleles combine into genotypes. And if we have a population that mates randomly, then these genotypes are formed by random combination. So if we have two alleles, they form diploid genotypes by combining randomly. And now if we have a population represented here by blue, dark blue dots and light blue dots, then both of these populations would be in Hardy-Weinberg equilibrium, meaning that the genotypes are formed by random combination. Now, if we have a mixture of these, mixture of samples from these two populations, then this group of samples will not be in Hardy-Weinberg equilibrium. If we can devise methods that will sort the samples in this mixture into the two original populations, then we will recover Hardy-Weinberg equilibrium, and this method would help us recover the population structure. And these sort of methods are nice in this way that they can, when principal components analysis will show you, let's say two or three dimensions in your data set that in one plot, depending on if you do two-dimensional or three-dimensional plots. These structure-like methods will give you more dimensions in one plot, as shown in the colorful plot below, where again we have worldwide populations from Africa all the way to Papua New Guinea, and the colors represent different genetic components. But genomes are not passed on as piles of nucleotides. They are actually linear molecules, and hence we actually have the genetic diversity is organized into haplotypes, which is basically arrays of variants that are that will be shared between individuals or between genomes. And you can use an approach called a copying algorithm. You can have a bunch of reference genomes, and then you have your study genome or test genome, and you can in the lower panel you can start walking along the genome and see from from which other genomes you can copy that stretch of DNA. And then we can see that in the lower panel, I do miss the pointer. We can start walking, and we see that we have an A and we have a G. And then from the reference set of genomes, we can see that we can only copy this stretch from population 1. There's one genome that also has A, G. Then we move further on, we see an A. And this is a point where we have to start copying from another genome, because the genome we copied before has a T in that position. So in this sense, we can basically paint the test genome according to our reference genomes. And in this way, we can, for example, study the effect of recent admixture, which includes for example, in case of Americas, the immigration of European ancestry people, and later, very soon after actually, also the African ancestry through slave trade. And we can see how in different regions of Southern and Northern America, where which ancestries are predominant. Now, natural selection, as I said, when let's say humans colonized the globe, we had to adapt to many different environments. But what is natural selection? Selection is actually just change of allele frequency, which is faster than would be possible through random processes. And now we can think of ways how to detect this. Of course, ideally is if we use ancient DNA, we measure or observe an allele frequency, let's say 10,000 years ago, and look what is the frequency today, and we have actual data. But in most cases, well, ancient DNA is there's not enough of it, let's say. So, but what happens if an allele increases frequency in the population very quickly? What happens is what is called selective sweep. So on the left panel, this red dot is a positive mutation that gives advantage to that to carrier of that mutation. And now this mutation spreads through the population, as in the plots to your right. But what happens is that not only this mutation increases in frequency, but also the neutral variation that is adjacent, which is close to this positive mutation. And what this means is that all other neutral variation that is in the vicinity of this mutation under selection will be lost in the population. So the sign of quick natural selection, positive selection, is a decrease of diversity along the genome around this position. So we can detect these selective sweeps, and for example, one such example of recent natural selection due to livelihood or what people do is example of Bajau sea nomads who have been diving for livelihood for they say thousands of years. We don't know the thousands, but a long time. And they have actually adapted for diving. Quite like sea otters, their spleens are much bigger than other human spleens. And spleen is important for diving because it holds a reservoir of oxygen-rich blood, which is very helpful if you dive and want to keep your breath for a long time. Okay, and just in the last minute, I'll also mention how culture can can change the genetic diversity. When we think of a coalescent process, which we actually saw in Johan's talk, a similar process, then when we go back in time, when if each let's say organism has a chance to either reproduce, give 1, 2, or 0 offsprings, then quite soon we will end up with one ancestor to all contemporary beings. But this will happen faster if we have a population bottleneck or just a smaller population size along the way, because just fewer lineages go through this bottleneck. So, which this means is that population size dynamics through history change the genetic diversity. And when we study this, we can, for example, see that around 5,000 to 10,000 years ago, there was a huge drop in male effective population size, meaning that very few males had offspring, but then the ones who had many offsprings, which in the female case is not observed and largely also not possible. But and this is how we interpret these results was that this is the time when agriculture was introduced or was becoming the main mode of subsistence. And what happens with agriculture is that you get wealth. You produce food, and then you have a big pile of it, and you have to guard it, and who guards it is the king, and gets a chance to reproduce more. So with that, I'll end. This is Institute of Genomics, most of us anyway, and thanks.
Yeah, thank you very much, Mike, indeed. Well, this documentary you have just seen, this is one of the leading labs in the world in population genetics, actually. I proudly can state. So, well, as you see, that this is not just simply genetics. It's related to very many problems, migration and cultures, as you mentioned, and finally. Are there any questions? Again, short questions first. If not, then thank you very much indeed, Mike, for a very interesting talk. And now, ladies and gentlemen, we continue. Well, much is spoken about energy for a sustainable development, but it's not only the problem of energy, it's also the problem of natural resources, and water is without any doubt some resource what is really inevitably needed for the society. So the next speaker is Dr. Jūratė Kravčiuvienė from Lithuania. She's from the Lithuanian Energy Institute and a member of the Lithuanian Academy of Sciences. So Jūratė, the floor is yours now. You're welcome.
Dear colleagues, at first I want to thank for invitation to this very interesting conference. And my topic is related with changes of water resources in the past and in future. And it is a very similar topic if I look to the presentation of my colleagues. In this presentation, it will be the main aspect related with hydrological regionalization of the territory of the Baltic states, also the changes of river runoff in the past, climate scenarios and hydrological modeling, changes of river runoff in future, and changes of hydrological regime and water ecosystem. The information in this presentation is related with 3 projects. At first, it is Lithuanian National Program, Sustainability of Agroforest and Water Ecosystem, and 2 cost activities. The river runoff is the product of climate, and the formation of river is related with the meteorological factors and also with physical geographical conditions. And there are some examples, the difference of characteristics in the Baltic states. The area of Baltic countries is not big, but we have a very big difference. For example, precipitation. Near the Baltic Sea, in the Latvia-Lithuania west part, we have big precipitation amount, and in the continental part, the precipitation amount is less. Very big differences in duration of snow cover. In the north of Estonia, the duration of the snow cover it is 160 days, and near the Baltic Sea, in the west of Latvia, only 70 days. And the formation, the main formation sources of the river runoff is precipitation, snow melting, and groundwater. And also it is a very big difference related with feeding sources. In the west part of Lithuania and Latvia, main sources of river feeding is precipitation. And in the continental part, it is the main sources is groundwater. So using all information and cluster analysis, we did regionalization of Baltic states, and one region is related with maritime climate. It is Lithuania, Latvia, west part. Another region, it is part related with continental climate influence. It is southeast of all countries, and it is transition region between these 2 regions. And this classification is necessary for the task that the river behavior in these areas are very similar. So it is useful to know which river are in which area. A very important part, it is hydrological extremes, floods and low flow. And for example, it is a map with the trends of flood discharges in Europe. And you could see Baltic states, we have negative trends of the flood discharges. It means that the floods in this region is going less and less. But there are another region in Europe with the positive trends. And it means that there are many flash big floods in summer and autumn time. Another trends are in the winter season. In our Baltic states, we could see that the runoff in the winter season increased. It means that spring floods is coming yearly and yearly, and now sometimes it is not spring floods, but winter time floods. In Lithuania, we have very long time series of water discharge and levels in the Nemunas River. So we have very long data, more than 200 years, and we could investigate this time series. We found cycle variation in long time series. It is wet and dry season, And also we could see the decreasing tendency of the average runoff in Nemunas River, and also the negative trend of the low flow. This is a little bit strange picture. It is changes of spring maximum discharges in the Nemunas River, and we have the break point in 1960. Why? It is two reasons. Because in this year, the Kaunas hydropower plant was constructed, and this Kaunas reservoir could have influence on the floods below plants. And also we have influence of climate warming from 1962. I spoke about changes of runoff in the past according to the measurements data, but what will be in the future with water resources? So at first we have to know to select the climate scenarios and climate models. After that, we have extract time series of temperature and precipitation for the forecasting period for the 21st century. And after that, we have hydrological model, and we could calculate daily water discharge in the future. And if we have daily discharge and water temperature, we could speak about the influence of these factors to the fish community or another task. That area, we selected 7 rivers. So there are many discussions do a climate change in our society. There are optimists and pessimists about this thing. But we have 2 pictures. It is reconstructed temperature time series 2000 years. And you could clearly see the decreasing of temperature. And it is time series from 8 to 60 years. And again, from the 1960, we again have very increasing trend of temperature. Anomaly, it is about 1 degree in average. So it is a very big anomaly. What will be in future? Many institutions are working with climate scenarios, and we have no RCP climate scenarios with the very big uncertainty. If we have optimistic scenarios, 2.6, the temperature could increase by 1 degree in the to the end of the century. But if we will have worst scenarios, 8.5, the temperature could increase to 5 degrees. So it is a very big uncertainty, and we are calculated uncertainties, things and so on, but we don't know in what way our society will go. So about hydrological modeling, we used HPV hydrological model and input data geographical information, observed temperature and precipitation, and also we used measured water discharges for calibration of model. When we got prepared hydrological model, we used data time series of precipitation and temperature, and we could model water discharge in the future. Calibration of model is very important. You could see this observed runoff and simulated runoff, and so I thought that this relation would be very close. what about future projections of air temperature and precipitation? It is the data of Kaunas Meteorological Station. So in the near future, the temperature could increase to 2 degrees according to different scenarios. But in the end of the century, you could see that color. It is the worst scenarios. So the temperature could increase to 6 degrees. So it is very, very big increasing. Precipitation changes has different character, but in summer times, it is expected that the precipitation amount will be smaller, and it is not good for the rivers. It could be hydrological drought. And it is projection of river runoff in different hydrological regions. The smaller changes is in the river near the Baltic Sea, in the west part. And the black line, it is the reference hydrograph, changes of water discharge per year. And the blue line, it is the hydrograph in the near future. And red line, it is hydrograph in the end of the century. So in some rivers, it will be very big changes. It will be no spring floods, and it will be very low discharge in the summertime. And water temperature is very important, not only for people, but for water ecosystem too. And you could see very good relation between air temperature and water temperature. And if we know what air temperature projections according to different scenarios, we could calculate water temperature projections in future too. And what about water temperature in the rivers? Again, black, it is reference period, and it is different scenarios, and it is worst scenarios, 8.5. And according to worst scenarios, water temperature in some summer months would increase to 4 degrees. So you could manage though it is not good situation for water ecosystem. And it is a small example from our works. You could see the river in the summer of 2015. It was a very dry season. It was less water and very hot water. And this is picture habitat accessibility for salmon spawning. And it means that in this situation, there are very small territory for good condition for salmon rivers. And it is the view in the next year, 16, and it is the better condition in the river. And very important result of our project, it is relation between water discharge, water temperature, and fish indices. There are two main groups of fish. In Lithuania, it is Carpian fishes who like more warm water, and salmon fishes who like cold water. And in the future, water discharge in the rivers will be lower. It is period in the end of the century. It is period in the near future. And water temperature will increase, especially in the end of the century. And the effect on the Carpian fishes index SP more than 16, you could see that the fishes, this index will increase, so it is good condition for this type fishes. But for salmon fishes, it is not good condition, and salmon fishes in some rivers could disappear due to that condition, due to water temperature and water runoff. And my conclusion, the main conclusion, that we expect significant decrease of spring flood discharges, also significant decrease of low flows of summer period, the dramatic increase of river water temperature. And this abiotic factors could have very negative aspects for the living conditions of water ecosystems. Thank you for attention.
Thank you, Rata, very much for the very interesting talk, because it's not, well, as I understand, water means that it's actually it's a result of the climate changes. It affects fisheries, it affects agriculture and so on, and this is really important for all the, not only for the Baltic countries, well, you have got the examples, but all over the world. So thank you very much indeed. Are there any short questions? No? Then, oh, yes.
Professor, I have a question. Sorry, probably this is not very professional to you, but the warming of the Baltic Sea waters and also our continental waters do stimulate also the increase of different invasive forms of fish. How do you may prognostic the existence of our local Baltic Sea and Baltic inland and water fish and their resistance against the invasive kind of fish?
Yes, you are thank you for very interesting question. You are very right. The invasion fishes are very popular, not in good, in good sense. In Lithuania, near shore too, we have some species which have influenced the water ecosystem condition, and it is a very bad situation. So from one side we have invasion species, and from another side the climate change affects the Baltic Sea water temperature too. And in many times for invasion people, it is better situation, better living condition than for our usual species of the fish.
Yeah, thank you very much. I think that we shall thank you.
Thank you.
And now we come to the last talk. Actually, we have been, all the talks were about the future and about the predictions and all that. But actually, we have to sometimes also to look back to understand how mathematics has been used a long time ago already. And the last speaker is from the Technical University of Braunschweig. It's Professor Thomas Sonar from the Institute of Partial Differential Equations, and he's a professor of technomathematics. And the problem is about the navigation and the history of navigation. So Thomas, the floor is yours now.
Thank you very much. Is everything okay with my audio? Can you hear me?
That's right.
Okay. So this is a talk on table making, actually. And I'm going to start to describe the situation in the 15th to the 16th century. Astronavigation was practiced and known in Spain and in Portugal. England was a country very much behind. There were so-called rudders in use, very primitive manuals. Pierre Gassi, a French navigator in 1480, design such rudders which were used by the English. Only the Channel and the Biscaya were included. 1541, the Rudder of the Sea by some Richard Prout occurred, which was the Gassi manual plus some English waters. Elementary mathematics was almost completely unknown in England at that time. In Spain, the Arte de Navegar by Martin Cortés was in use. It was written, or it was published in 1551, and that was the standard source of Spanish mariners. And of course, the English and the Spanish did not go well together. So some English ships conquered Spanish ships, and they brought with them the Arte de Navegar. And in 1561, the first English translation occurred as the art of navigation. A few words to navigation itself. It is clear that only computation of latitude was possible at that time. Longitude could only be determined in the 18th century by the invention of an exact clock. And either you could look at night at the height of Polaris. I'm not going to talk about this method. Or you could measure the height of the sun. So we are concerned here with the navigation concerning the height of the sun. You know that our ecliptic is inclined by about 23.5 degrees. That was known by the Greeks already. You have to determine your time at noon, and then you measure the height of the sun at noon, which is the angle alpha in my little sketch, which you see below. It seems to be a bit complicated, but in fact it isn't. It is very, very simple geometry here. And it turns out what you really need to determine your latitude is the so-called declination of the sun, which is the angle delta. If you would know the angle delta and you would measure the angle alpha, then you could determine your latitude beta simply by subtracting alpha minus delta from 90 degrees. So this is what a navigator on board a ship had to do to determine his latitude. Very much in use was the astrolabe to measure the sun's height at noon, but this was very rough only. Think of standing on board of such a ship 25 meters in length and you are out on the Atlantic. You will never get a correct measurement with this method, but nevertheless this was used. The basis of all the tables to determine things like the declination are the Alphonsine tables. The start of the data used was the 1st of January 1252. That was the coronation day of Alfonso the 10th of Castile. And these tables contained the position of sun, moon, planets relative to fixed stars. This old corpus, which is extinct nowadays, was the basis of all what people like Cortés and others did in navigation. Here I have a copy of one of the beautiful pages of a very old edition from Paris. The problem with these tables is we don't have authentic Alphonsinian tables. The contiguous corpus is extinct. The oldest reference we have is what is called the Parisian corpus some 100 years later. There are neither translations into English or German nor critical editions in these languages, so that very few people can handle the Parisian corpus. I know exactly one person who is able to deal with that table, and this is Mathieu Husson in Paris. I can't. I simply can't. I don't understand enough of these tables. The Alphonsine tables were the most widely used in Europe up until the 17th century. Many people say that Copernicus was a revolution, but in terms of astronomers and navigators, it was not a revolution. These old tables were used long after Copernicus published his famous book. In 1551, there were new tables produced by Erasmus Reinhold, the Prutenic or Prussian tables. They used Copernican theory, but practically these tables were not used outside of the German lands. And the end of the Alphonsinian tables came only in 1627, when Kepler published the Rudolphinian tables. What is the structure of the tables? Let us at least take a spot look on the structure of the tables. An important thing is the true place of the sun. What is that? The true place of the sun is the sun's latitude lambda on 365 days in the course of 1 year. And then you need the equation of the sun. When you have both, the true sun's latitude is the true place plus what comes out of this equation, and this depends on the year. And the declination delta then follows from the true sun's latitude. So this is the principal structure. So you need the declination. You are standing on board a ship made of wood, 25 meters in length, and you have to compute or determine your latitude. So you use Cortes' art of navigation. You need 3 different tables. Table 1 is the table of the true place of the sun. And I'm citing here from the English translation of 1561. The true place of the sun is a point or prick in the zodiac, which is thus found, that drawing a right line from the center of the world to the center of the sun, and carrying the same continually right firth unto the zodiac where this line sheweth or toucheth, that is the true place of the sun. Here is the table for the true place of the sun, table number 1. And we take as an example the 22nd of February. So this is February, as it is told by this table, February. We are in Aquarius, seemingly. This will change. There we have on the left, in the left column, you see the number of the day. So the 22nd. You get the reading 12:58. So this is 12 degree 58 minutes. But be careful, there is the sign of Pisces there. So what happened is, what happens is the sun has changed on the 10th of February from Aquarius to Pisces. And this you have to remember when you take the reading from this table. Then to the, this is another citation, then to the degrees and minutes which you shall find, you shall add the equation that is directly of the year in which you are or seek to know. And this shall you seek in the table of equations, which is after this. And then there is a description of what you have to do if you have a leap year. And the rule gives you no leap year, subtract 1 degree. So if you don't have a leap year, subtract 1 degree. This is the table of the equation of the sun. This is the second table. We take as an example the year 1568. This is a leap year. So you have in the left-hand column, you look up the number of the year. It's 1568. You get a 25 there. This means 0 degree 25 minutes. And you add that to the result you got from the first table. And you get 13 degree 23 minutes. The 22nd of February 1568, we know already the sun is in the zodiac sign Pisces. So you have the third table. Pisces is in the lower part of the table. That means you have to read this third table from bottom to top. We are in this column. We have 13 degree 23 minutes. This is between 13 degree and 14 degree. You have that on the right, in the right hand column, 13 degree and 14 degree. So you know where you are. This is the entry which interests us. 13 degree gives 6 degree 42 minutes. 14 degree gives 6 degree and 19 minutes. And if you do linear interpolation, that was mathematically possible. Even in England, you get a declination of 6 degree 33 minutes. Pooh. So that was what you had to do if you had nothing but the tables of Martin Cortés on board. This was at the time much too complicated, as was said in one book, for ordinary mariners. You would have to take a PhD with you to do this lookup in the table. So fairly early, the desire was to get a method with much less table to use. And William Bourne is the one who started the process. He lived from about 1535 to 1582. And he had a wonderful career, I think. He was an innkeeper, he was a gunner, and he was a mathematician, and he came from Gravesend. And Gravesend was a very important harbor in these days. He learned mathematics from Robert Record's books, as did nearly all of the English who were interested in mathematics at this time. Gravesend was an active harbor, I said that. He was interested in practical navigation, and he intensely studied the art of navigation by Cortés. And in 1567, he published his book, Almanac and Prognostication, from which we have not a single copy. So nothing is preserved from the first edition. But in 1571, there was a second edition, and I show you here the title page. You need one declination table instead of 3, and this was a very big leap forward. And as an example, 22nd of February 1568, you get from this table 6 degree 52 minutes, and there is a difference to Cortés in 19 minutes, which means 34 nautical miles. This is the table. Since one is not allowed to copy these books, there was an attempt to set this book new in a typesetting language, and here you see the table. 22nd February 1568, so you have the almanac for 3 years. 1568 is here. We are in February. We are on the 22nd of February, and there you can read 6 degree 52 minutes, which was a gigantic leap forward. Born in 1574, published a regimen for the sea containing most profitable rules, mathematical experiences, and perfect knowledge of navigation for all coasts and countries, most needful and necessary for all seafaring men and travelers as pilots, mariners, merchants, etc. And this became the standard reference of the following decades in England. Navigation was popularized by many men of the day. The most famous one, I think, is Thomas Blundell. Blundell was a major popularizer of sciences, and in 1594, he published his exercises containing 6 treatises. There are a lot of later editions, and then it goes up to 8 treatises, I think. Somehow at the end of the 16th century, these exercises contained 8 treatises. It was written for the education of young gentlemen, and it was thought at these times that young gentlemen should be able to understand a little bit about navigation. And in this Blundevil's book, there is this declination table, and the declination in Blundevil's book is known if the position of the sun in the zodiac is known. Then comes Thomas Harriot. Without saying, the greatest English mathematician of his days. He was the private mathematician to Sir Walter Raleigh. He is one of the inventors of the telescope. He accompanied a famous expedition to Virginia, but that is not of interest to us. He was a great astronomer. He measured the inclination angle of the ecliptic. He includes the parallax, and he includes the refraction of the atmosphere in his regiment of the sun, 1593 to 96. These were the most accurate tables at the time. And he invented the difference column in red, printed in red, for redundancy. So you had a means of checking whether there is a printing error in your table. The perfection, the last point I want to make, the perfection came with Edward Wright, a cartographer, a mariner, and a very well-known applied mathematician of his age. He is known for his rigorous construction of the Mercator map, the first man in England to understand the intrinsicalities of the Mercator map, and he was a member of a well-known and famous circle around Henry Briggs at Gresham College in London. He published in 1599 certain errors in navigation arising either of the ordinary erroneous making or using of the sea chart, compass, cross-staff, and tables of declination of the sun, and fixed stars detected and corrected. So he detected and corrected a lot. This book experienced many new editions. Here is a title page of, I don't know which edition this really is, but it's 1657 already. And here is the declination table, one table as was proposed by William Bourne, and you have the difference column. He took that from Thomas Harriot to get a bit of redundancy into the tables and to make the use a little bit more accurate to use. The computation of the tables, we are told in the preface of Edward Wright's book, was done by Henry Briggs, the inventor of the logarithms. Okay, final thoughts. Wright's certain errors is the culmination of a development which went very fast. 1561, we had the art of navigation in England, Cortes' book, and 1599 was certain errors by Edward Wright. So within 30 years, these techniques of navigation were very developed, very much developed in England. And the remaining question, which I am after for 30 years or so, is: was there a connection between Briggs, Wright, and Harriot? It seems to be they cite themselves in their books, but we simply don't know. Okay, thank you very much for your attention.
Much, Thomas. Indeed, it's always so interesting to listen about the history of mathematics and the ideas that were maybe 300 or 400 years ago, and now it's how it started, like this in Gresham College, which was actually the predecessor of the Royal Society, and then still it exists. So, thank you very much. Are there any question about the navigation? Probably not. So thank you, Thomas, once again. And this is was the last talk of our meeting. Now, if you allow me a couple of words. Does it work here? Yeah. Yeah. Just some final words. So that we had 3 sessions. And we suddenly, the main message to take home is that the mathematics is really the powerful tool to understand the world. The problem is the prediction. But as we know, the prediction is always very complicated, especially when it concerns future. So it was, like Niels Bohr said, more than 100 years ago. But nevertheless, we know that there is the powerful tool, mathematics. We know the time series that was also explained yesterday. We know universality of several processes, phenomena, and so on. What is important is bioinformatics, quantum computing today, and natural resources. So these all actually need mathematics. And the point is actually that also that was touched yesterday during the first session. Yeah, well, we know quite a lot from various fields, but what about the policymakers? Could they get this knowledge? And as we know, for example, about the spread of pandemics was really known, well, many years ago, actually. For example, Dirk Helbing from ETH has explained, when was it, in the 10 or 15 years ago, about the spread of viruses. But nevertheless, that what we met now is, or faced now is really the collapse of many systems. So that mathematics, exactly as it was also explained by Greta Fischer and the colleague from Latvia and Lithuania, this is the tool to understand it. We certainly, what is important that the next year, actually, it's for the UNESCO, it's called the International Year of Basic Sciences for Sustainable Development. I think that our conference was actually the first step towards it. The Estonian Academy of Sciences has also signed an agreement to support these activities, and quite probably next year we shall have some more into this scheme. And now, as a chair of the organizing committee, I would like to thank all the speakers from today's meeting and the session or the yesterday's sessions. And I would like to also thank the staff of the Estonian Academy of Sciences for all this technical help and for all organizing the starting from the coffee breaks and the dinner and there is something coming on there. So I would like to thank you very much, and now I invite Tarmo Soomere, the president of the Estonian Academy of Sciences, just to say the final words. Tarmo.
Also, many thanks to everybody from my side as well. So what we have seen here, to my understanding, it has been a extremely interesting firework of ideas, concepts, results, and strictly speaking, not applied science. Actually, there is no division of science into fundamental and applied science. There is division into excellent and not excellent science. And some people are saying that if you do only applied science, there will be soon no science to apply. What we have seen was excellent science, ready for applications, and in many occasions with demonstrated capacity and high performance to serve for the benefit of society. And this is what people usually call applied science. It has been now 86 years of intellectual cooperation between the Baltic states, okay, with 50-year-long period of hibernation. After that, we have gathered now 10 times and have issued total 36 medals. And it's clear that we wish to continue cooperation. I think it is extremely necessary, having in mind things what Jüri Engelbrecht just mentioned. It's becoming a kind of a cliché to say that the competitiveness of countries and nations and regions and continents, and also the prosperity of countries, and of course quality of life, it directly depends on the capacity of the policy-making branch of the country to extract the best knowledge from the most educated and most competent people in the country. And not only to extract this, it is said that a good government is not who is wishing to make people happy. A good government is who knows how to do that. And as has said foreign member of Estonian Academy of Sciences, Helmut Schmarz, without science we can't build future which is worth of living. There are some obligations of scientists who belong to the academies. In some sense, or in a very rough way, former Academy President Karl Rebane has formulated it in a sort of negative manner, saying that the ultimate goal of academies is to avoid stupid strategic decisions. And this is pretty much true, and it's even more true than we could imagine. If we think about different ladder steps in science, and doctoral degree is a clear quality mark, certification of a person being more or less independently retrieve new and important knowledge and communicate that to others. This is the point of science. A professor is a top scientist, established researcher with long CV, with ability to teach, is the first component of being professor. And the question is, what is the quality of being academy member? What is extra? What kind of distinguishing it is? Is it just a hall of fame where you are hanging forever? No, told our academy member Anto Raukas, who passed away just a few months ago. He told that academy member is a cultural phenomenon. It must be much wider in understanding of the world. It must be much sharper when it's about passing or channeling knowledge to policy making. We are in extremely changing times, and one of the features of these times is that we have to make sure for society that science is important. Not only in terms of results. Society doesn't ask us how many theorems we have proved or how many equations we have solved. Society asks how we make their life better. And the component is now, which is being worked out in all European countries and in many countries of the world, this is giving the best science advice for making policy, for political decisions at all levels of society. Not only education, not only higher education, not only research, not only economy, but running the country wisely. It is clear that a small country cannot support all fields of science on excellent level. In Estonia, the number of scientists is smaller than the number of lines in classification of science. And it's pretty much the same for all countries with population below 20 million people. So we are strong only together, supporting each other and offering complementary knowledge. And there is another pressure of time, which has been mentioned several times during those 2 days. Science is no more a passive observer. Scientists are no more just those who describe what will happen. They even no more people who predict what will happen. It's now time for science to be not reactive, but proactive. It is now time when smart people possess so much knowledge and experience and possibilities that it is possible to change the world. And it is our choice whether we do it for blessing or curse. Those things will be discussed in detail in the follow-up of this conference, which will be the meeting of presidents of 5 academies of science. But returning to science, and where I see one massive change in understanding of science. Understanding as such is a category which has the potential to change the world. If you understand things, then you normally act differently. Pretty much over the last almost 60 years, the world has been divided into bits, 0 and 1. And every computation has been performed on that level. This is very poor description of our world. What we see is now the emerging much more rich and complex description, and its first swallow is the quantum computing, where you are not limited to 0 and 1. So the world is becoming colorful again, and this is a nice message. Thank you very much for your investment into this conference.
Now, ladies and gentlemen, the conference is closed. The lunch is served in the neighboring room here, and the presidents will meet shortly in another room over there. So thank you very much, and have a nice lunch, and have a nice journey home who have been coming to Tallinn. So thank you.